The scene opens in a lecture hall, filled with eager medical students. Professor, your name, stands at the front of the room, ready to deliver a captivating lecture on the intricacies of gene expression. The lecture is about to begin. Professor, your name, good morning, everyone. Today, we will embark on a comprehensive journey into the realm of gene expression and its underlying mechanisms. Gene expression is a fundamental process that allows cells to transcribe and translate genetic information into functional proteins, ultimately shaping the biological processes that define life itself. In this lecture, we will explore 12 key topics that play critical roles in controlling gene expression regulation. So without further ado, let's dive right in. Gene expression begins with the recruitment of RNA polymerase II to gene promoters. However, this initial interaction may lead to a pause state, where the polymerase stalls before entering the elongation phase. Promoter proximal pausing is crucial for precise control of gene expression levels, allowing rapid response to signaling cues. Various factors, such as NELF and ZIF, regulate and release paused polymerases for efficient transcript elongation. Enhancers are DNA regions that can activate or enhance gene expression from a distance. Through long-range chromatin looping facilitated by protein complexes like mediator and cohesin, enhancers can interact with specific promoters. These interactions bring the transcriptional machinery closer to gene promoters, leading to increased gene expression. Disruptions in enhancer-promoter interactions can result in diseases like cancer or developmental disorders. Gene expression is influenced by the accessibility of DNA regions. Chromatin remodeling complexes, such as SWI/SNF and ISWI, utilize ATP-dependent mechanisms to modify chromatin structure. By sliding or ejecting nucleosomes, these complexes can expose or conceal gene regulatory elements, ultimately affecting gene expression levels. Transcriptional co-activators and co-repressors serve as essential intermediaries between transcription factors and the basal transcriptional machinery. Co-activators, like CBP or P300, enhance gene expression by modifying chromatin and facilitating the assembly of the transcriptional complex. Conversely, co-repressors, such as NCR/SMART, inhibit gene expression by actively suppressing transcriptional activity. Epigenetic modifications, including DNA methylation and histone modifications, play crucial roles in gene expression regulation. DNA methylation, typically associated with gene silencing, involves the addition of a methyl group to cytosine residues. Conversely, histone modifications, such as acetylation or methylation, can either activate or repress gene expression by altering chromatin structure and accessibility. Non-coding RNAs, NCRNAs, have emerged as key players in gene expression regulation. Long non-coding RNAs, LNCRNAs, microRNAs, miRNAs, and small interfering RNAs, siRNAs, can modulate gene expression at various levels. For example, miRNAs can bind to specific messenger RNAs, mRNAs, leading to their degradation or translational repression, thereby regulating gene expression in a post-transcriptional manner. Alternative splicing allows for the production of multiple mRNA isoforms from a single gene, vastly expanding proteome diversity. By selectively including or excluding specific exons, alternative splicing can generate protein variants with distinct functions or regulatory properties. Dysregulation of alternative splicing has been implicated in various diseases, highlighting its importance in proper gene expression regulation. After transcription, mRNA stability and degradation play critical roles in determining gene expression levels. Regulatory elements within the mRNA sequence can influence its stability, allowing for fine-tuning of gene expression. Additionally, protein complexes, such as the RNA-induced silencing complex, RISC, and decapping enzymes, control mRNA degradation rates, ensuring precise gene expression control. Post-transcriptional modifications, such as capping, polydenylation, and RNA editing, contribute to gene expression regulation. 5-capping protects the mRNA from degradation and facilitates its translation, while polydenylation affects mRNA stability and export. RNA editing, on the other hand, alters the nucleotide sequence of transcripts, resulting in functional changes at the protein level. Gene expression control extends beyond transcriptional regulation. Translational regulation allows cells to fine-tune protein production by affecting mRNA translation rates. Regulatory elements within the mRNA sequence or the presence of specific proteins, such as miRNAs or IFs, can influence translation initiation, elongation, or termination, ultimately determining the amounts and types of proteins synthesized. Feedback loops and autoregulation mechanisms provide additional layers of control in gene expression networks. Transcription factors or signaling molecules can regulate their own expression through feedback loops. This enables cells to maintain homeostasis and respond dynamically to ever-changing environmental cues, ensuring appropriate gene expression patterns. Dysregulation of gene expression lies at the core of numerous diseases, ranging from genetic disorders to cancer. Understanding the mechanisms underlying gene expression dysregulation can shed light on disease pathogenesis and potentially lead to the development of targeted therapies. Examples include aberrant DNA methylation patterns, altered splicing events, or disturbances in regulatory RNA networks. Gene expression regulation is an intricate and multifaceted process that involves a multitude of interdependent mechanisms. From promoter proximal pausing to disease-related dysregulation, each aspect we explored today plays a crucial role in ensuring precise and controlled gene expression. By unraveling the complexities of gene expression, we can gain invaluable insights into disease mechanisms and potentially pave the way for novel therapeutic interventions. Professor, your name, thank you for your attentiveness throughout this lecture. I hope you found it enlightening and that it deepened your understanding of gene expression regulation. If you have any questions, feel free to ask.